Good morning, Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay Sir, Sister Nivedita University, respected delegates, faculty members, and dear students. Greetings of the day. It is with great pleasure and honor that I extend a warm welcome to National Symposium SNU Bio-Talk 2024. This year, our theme is Plant Biology in the Post-Genomic Era, Strategies for Crops and Mankind. As we gather here today, we are all united by our shared commitment to advancing knowledge, fostering innovation, and addressing the complex challenges that confront our world. The symposium, organized by the Department of Biotechnology and Department of Microbiology, Sister Nivedita University, promises to be a melting pot of ideas where the brightest minds come together to share insights, discuss breakthroughs, and forge collaborations that will shape the future of our scientific endeavors. Over the next 2 days, we will embark on a journey through a meticulously curated program featuring eminent speakers, interactive sessions, and engaging discussions. This platform is not just a testament to our collective quest for understanding, but also a tribute to the spirit of inquiry and the relentless pursuit of excellence that characterizes our scientific community. I wish you all an enriching and enjoyable experience. May the discussions and deliberations over the next 2 days inspire innovative ideas and contribute significantly to the advancement of science and society. Once again, a very warm welcome to the Scientific Symposium SNU Bio-Talk 2024. To commence with, may I kindly invite our Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay Sir, Professor Arun Lahiri Mojumdar Sir, our conveners, Dr. Surjit Singh, Dr. Siddhartha Dotto, and Dr. Shori Roy, and all other faculties to take part in the symbolic act of nurturing and growth. Kindly join us in the ceremonial watering of plants. Thank you, sir. Thank you. May I now have the honor of inviting our Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay Sir, to the podium to deliver the welcome address.
Very good morning to my dear students, the faculty members of Sister Nivedita University, Department of Microbiology and Biotechnology, our very, very honored guests. For me, they are my good friends. Just to meet with all of them is something which I'm feeling so nostalgic. So, my dear students, the reason why we started this BioTalk is to identify the focused areas in the microbiology and biotechnology for the students to understand what sort of research is going on and to develop their inquisitiveness, their interest, and they can actually think about what and where they can fit in. And that's why we are starting with this time with plant biology. And how we're looking at the plant biology. This is, if you remember that in the COVID time, if you look at in general the survey report from the economic times, they always said that India, because of its agroeconomy, we are not being affected so much like many other countries. And this agroeconomy is something which is very, very important. And when you are talking about the sustainability, these things are becoming very important. Now, for the students of microbiology, molecular biology, biotechnology, they should actually get some idea about that what is actually where the plant biology stands in the post-genomic era. And we try to focus it on the strategies for crops. And always in every symposium, whether this is biotechnology or any other subject area, we want to keep the society, the mankind, to relate our students that they have to contribute whatever they're learning, whatever they're doing to the society itself. So that's why our title is Plant Biology in the Post-Genomic Era, Strategies for Crops and Mankind. Now, just for the speakers and the people who are coming from the other different parts, I wanted to mention that Sister Nivedita University, it started its journey in 2018. So 2018, when it started, it's only 200 students, and that's also under the mentorship and guidance of our founder, Vice Chancellor, Professor Ashok Ranjan Thakur, who is over here. I'd just like everybody to give a very good big hand for him, because it's his vision. He tried to develop the different departments in a way where the cross-disciplinary understanding are going to be the major thing in this university. You know that it's not during the NEP. Much before NEP, in our university, we started the practice of how the students of all different subject areas, they can mingle with each other. They can interact with each other. They can do, and also we have given a lot of emphasis on extracurricular activities. And extracurricular activities means understanding the importance of not only their subject areas, but the subject areas of other departments also. And that's why one of the major thing in our university is the year-long extracurricular activities. There are so many. In fact, just today we are having 5 different programs in 5 different areas going on. And in one case, we are also hosting the All India Psychiatric Nurses Conference. You know, we have a nursing department, and the psychiatric nurses from all over India, 400 people are going to assemble over here. So you can easily, though we are very small, our space is very little, but our ambition is quite a bit, and we are taking this challenge. And whoever you are taking the photographs and everything, and all the live streaming, the YouTube, social media, everything was done by our students. They are from the Department of Mass Communication and Journalism. So we are really thankful for them, that because this is also a learning process for them, and they are also take it as their internship. So in that way, each and everything we wanted to utilize in the way so that it will help the students at least to have a mindset which is broader than a narrow one. And from that end, as I mentioned to you, 200 students it started, and right now we have 8,000 students. So in these 5 years, it has grown in an exponential fashion, and that is one of the challenge for us also. But it is not only the Department of Molecular Biology, Biotechnology, Microbiology, they are doing this type of specific seminars and others, academic, different types of academic programs. All the departments are used to do that. And that is actually one of the what I could say is a signature of our university. With this brief introduction of our university, and I also want that yesterday Chancellor Sir personally met me and said he will try to come over here to meet with our all of you, but because you can easily understand there are 5 different programs going on, he has to also move to one place or the other, and that's why he will try. I don't know, in the throughout the day sometime he may come and join us. With these few words, again thanking specifically the organizers, the students, the faculty members of the department, as well as the support from the other departments for trying to make this symposium a real success. And this symposium is just to make some networking for our faculty members and the students also to do for their future work and research. Thank you. Namaskar.
Thank you, sir. May I now have the privilege to invite our founder, Vice Chancellor, and our mentor, Professor Ashok Ranjan Thakur, sir, to deliver his address.
Good morning to all of you, respected Vice Chancellor, the distinguished delegates to this conference, and my dear students and the faculty colleagues. Indeed, before you are sort of regaled with some of the most exciting kind of investigations that's going on in the plant biology segment at the international level by the most distinguished guests who have joined us in this 2-day program, it becomes a very irritating interlude for a person who is quite outside the subject to address you. So I'm not going to address. All that I'm going to remind you is that, as it was described by Noam Chomsky in his For Reasons of State, that it was Alexander von Humboldt who defined the structure of the university to be the place where the different aspects of the expressions of mind needs to meet. And Chomsky goes on to write that if you don't have one, create one. And I think this is best exemplified by the name given to our university in the name of Sister Nivedita, who was the mentor to the first biophysicist of the country, Sir Jagadish Chandra Bose. In fact, Sister actually edited some of his scientific writing, just as she did one for Okakura, the Japanese artist who came down to Kolkata in the early part of the 20th century. And she also gave an understanding to what the education policy could be in a Bombay talk. She started a club called Sesame Club in England, where Aldous Huxley, George Bernard Shaw, and others spoke. So, you know, we cannot claim to have the proud privilege of that heritage. All that we are trying to do is to aspire so that our young minds, both the young faculties as well as the students, can really sort of interact deeply with each other. As the Honorable Vice Chancellor has already mentioned to you, right from the beginning, this university has striven to make sure that our perception of understanding science, or nature to be precise, does not boil down to understanding it from the narrow divisions of physics, chemistry, or biology. because nature does not make those divisions. We, with our limited understanding, quite often try to parcel out different segments and try to understand it. And the best part is when we get into a holistic understanding and join them together. It becomes a very difficult task because it's very difficult to cross one's boundaries of discipline and move into the other. But there is a way out. The way out is very simple. Reach out to the friends across the discipline, and I think you can always dream some of the brightest dream that can uplift the mankind. The Vice Chancellor has already spoken to you about the need of understanding the present day plant biology. And since one of the basic understanding of how life came around in this planet almost evolved with the plants growing up at the microbial level, the archaea that first got evolved. It would be an interesting 2 days of very interactive understanding of different areas to which we shall be regaled. And I'm sure my dear students would look up to this opportunity in their life as one of the greatest boon that came their way. With these few words, I thank you all for having come down and to Calcutta with all its glories as well as its defects, and let's enjoy the next 2 days. Thank you.
Thank you, sir. With great pleasure, may I now invite Dean of Science, Sister Nivedita University, Professor Mala Mitro ma'am, to enlighten us with few words.
Very good morning to all of you present here, dignitaries, renowned scientists who have come over here from a different part of India. Very good morning, and again a very warm welcome from Sister Nivedita University. This symposium, which has been organized by the Department of Microbiology and Biotechnology, will be with all the faculty members, students, involvement of all the faculty members, students, and all the staffs of this department and of many staffs from other departments. This, I hope that it's going to be a very successful one in terms of collaboration and the networking and collaboration, and the almost lifetime opportunity for many students to listen to the scientists who has got repute of international standards, and we are really expecting that our collaborative effort with some of these scientists at least will be fruitful. At least our students and faculty members will have the opportunity to interact the first stage of the collaboration with these kind of symposiums. With I can remember that we were interacting, myself, Professor Thakur, and Professor Arun Lahiri Mojumdar is also present here. We were interacting with the young bunch of faculty members, which we are really privileged to have a very dynamic faculty members in our in this department. Actually, in all the departments of this Sister Nivedita, many of the departments of Sister Nivedita University. But since the symposium is organized by this department, let me focus on this. So I can still remember we were discussing different aspects, and there also we, with the guidance of obviously Professor Thakur, our Honorable Vice Chancellor Professor Dhupajyoti Chattopadhyay and Professor Arun Lahiri Mojumdar, we have tried to mix up the various disciplines which are not only engaged into this particular department, also with the other department of science, and also with all the other disciplines which we are privileged to have as this is a university. Like I'd like to emphasize that we have psychology, we have nutrition and dietetics. So all these departments, they have got a very keen inclination, keen overlap with microbiology, biotechnology. So there is a huge scope of growing for the young minds, whether be students, whether be research scholars or the faculty members, to grow and to go to different disciplines so that they can serve the mankind, which is very, very aptly used in the title of this symposium. So I would also not take much time from these. I know that next one is from Professor, our next keynote speaker, Professor Usha is here, and I am really grateful that we can meet so many renowned scientists here. So you will expect that our, just a few words, that our students and faculty members will get and will interact with the facility, with the opportunity which have been provided through this symposium, and then only this will be a very successful one. So looking forward to that, with that hope, let me again welcome all of you to this beautiful morning with this beautiful growing university. Thank you.
Thank you, ma'am. With great respect, may I now invite the distinguished INSA honorary scientist, Professor Arun Lahiri Mojumdar, sir, to bestow upon us with his esteemed perspective.
And this symposium is a brainchild of this person. And because of Professor Arun Lahiri Mojumdar, right after joining us, he started actually mentoring all our faculty members in such a way that you have to work in this way, work in that way. And you know that who are the conveners? The two conveners, Shori is also applying, but two conveners already received one DBT project, one DST project in this planned science. So that's what actually we were trying to have, that we have to have faculty members actively participating in different researches, and all these things are really being nurtured in such a way by our mentor, Professor Arun Lahiri Mojumdar. Arunda, please.
Thank you, Dhruv. I'm never comfortable in presence of the vice chancellors, deans, particularly the founder vice chancellor of this university. 2 years ago when I joined, I asked the vice chancellor what should be my job. I don't have any lab. I'm not having a research project here. So what he said is just mix up with the faculty members, young minds, which I never disliked. I always like to interact with the students, faculty members, never comfortable with directors and vice chancellors. But still, these two vice chancellors, the founder vice chancellors and the present vice chancellors, were known to us, known to me for a number of years, made me very comfortable here. And I'm glad that I didn't have to seek what should I do, what shouldn't I do. And the vice chancellor has mentioned. And like vice chancellors and directors, I cannot give you an extempore speech, so I thought that I better write it out. So what should I say? So this is what it is. Formally, good morning, everybody, and welcome to the National Symposium of the SNU Bio-Talk. Sorry, Shomshubro, I said biotech, it's Bio-Talk. The name was coined by him, the head of the biotechnology department. It's on plant biology and post-genomic era, strategies and crops and mankind. This university, as the Vice Chancellor has already mentioned, is only 5 years old. And you can see that in 5 years, whatever has been done by the two Vice Chancellors, former founder and the present Vice Chancellor. Now, the idea of organizing annual symposium termed SNU Bio-Talk was generated during discussions among the faculty members of the department and finally formalized in the departmental research committee of the university. Based on the expertise and interests of the current faculty members, 3 distinct areas were chosen for such annual events, namely plant molecular biology and biotechnology. No bias towards me, but it was chosen by them. Environmental sciences and eukaryotic and organismal biology with emphasis on ecology and evolution. The present symposium is the first of the proposed series and is expected to be continued with other areas in coming years. The symposium has been organized among 3 themes, and you can see that, namely plant cell morphogenesis and development, plant stress biology in the post-genomic landscape, and translational approaches in plant systems. In each, we have 1 keynote speaker, followed by talks by other scientists who contributed substantially in the thematic area. We are extremely happy that most of the scientists we approached have agreed to come and participate in this symposium. Thanks to all. We tried our best in organizing the program, but it's certain that there has been a lot of shortcomings in doing so and had scope to improve. There are several aspects of current plant biology research which you couldn't accommodate, but would have very much liked to do. Some of them are genome editing, artificial or synthetic plant genome, in line with the recent Piscometrium work, riboregulation of gene expression by riboswitches and RNA thermometer. Incidentally, the RNA thermometer has been discovered in algal chloroplasts very recently. A month ago, the paper was published. And a very important work on the computational biology and plant databases. I know there are scientists who are involved in plant databases, but we haven't been able to contribute much to the computational biology and genomics area as yet. Medicinal plants and plant drugs, organelle genome and their evolution. We hope we'll be able to make up for such deficits in the near future. Calcutta had been a center of plant biology research since the 60s, with active research groups in the universities of Calcutta, Kolani and Bardhaman, and research institutes like Bose Institute and the Indian Institute of Chemical Biology, formerly known as Indian Institute of Experimental Medicine, with notable contributions in plant cytogenetics and genetics, plant physiology, phytochemistry, and plant biochemistry and molecular biology, which took leading roles in formulating national educational and research directions in these disciplines. We believe that the young sister Nivedita University would also be able to contribute substantially in maintaining the tradition in days to come. We seek your good wishes, encouragement, and advice all through in our journey in achieving such goal. Thank you very much.
Thank you, sir, for your esteemed perspective. May I now request our program coordinator and assistant director, Department of Biotechnology and Department of Microbiology, Dr. Rajat Pal, to felicitate our Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay, sir. Second. Okay, felicitation. Felicitation. May I now request Dr. Rajat Pal to felicitate presentation. Okay. We don't have presentation. No. Okay. So we embark on a fascinating journey with our first session, dedicated to the exploration of plant cell morphogenesis and development. We will delve into critical discussions and presentations that shed light on the theme, setting the tone for a day of insightful learning and exchange. It is my distinct honor to introduce our session chair, Professor Moitri Dasgupta, ma'am. Professor Dasgupta is a renowned academician affiliated with the Department of Biochemistry at the University of Calcutta. Her research interests lies primarily in the field of symbiotic nitrogen fixation and the structure function of plant receptor kinases. Throughout her career, Professor Dasgupta has established a vibrant research program, contributing significantly to her field by training several PhD and postdoctoral researchers. Her commitment to advancing biochemistry and her contributions to the scientific community are evident from her extensive body of work, which includes numerous publications and citations. In addition to her academic qualifications, Professor Dasgupta has been honored with the prestigious J.C. Bose National Fellowship, marking her as a pivotal figure in the scientific community. The session will be co-chaired by our Honorable Vice Chancellor, Professor Dhrubajyoti Chattopadhyay, sir. Professor Chattopadhyay is a distinguished academician who has served as the Dean of Science, Faculty of Science at University of Calcutta, and Pro-Vice Chancellor, University of Calcutta. He was the director at the Center for Research in Nanoscience and Nanotechnology at University. Professor Chattopadhyay became the Guho Professor at Dr. B.C. Guho Center for Genetic Engineering and Biotechnology, University of Calcutta. He is the recipient of numerous awards, accolades, and distinctions. He was the Fellow of Indian Academy of Sciences, Fellow of West Bengal Academy of Sciences and Technology, to name a few. Professor Dhrubajyoti Chattopadhyay excels in both his academic pursuits and research, equally renowned for his pedagogical skills. His distinction as a scientist and educator underscores a career marked by significant contributions to science and a dedication to fostering the next generation of students and scholars. It is with great honor that I invite Professor Moitri Dasgupta, ma'am, and Professor Dhrubajyoti Chattopadhyay, sir, to grace the dais and commence the first session of today's symposium, Plant Cell Morphogenesis and Development. Thank you, sir. Thank you, ma'am.
Hello.
Good morning. Good morning, everybody. And where is Usha? Yeah, it's a very warm welcome to you. You sit there. Let me read out this, and then you come here. Okay? I'll call you. I'll call you. So don't hurry. So Professor Usha Vijayaraghavan is a friend of mine. That is the first thing, and then I'm going to read out this. Okay? So she is a professor in Department of Microbiology and Cell Biology and in Indian Institute of Science. I know her for last 2.5 decades, maybe. She did her PhD in Caltech in molecular biology and genetics. I think East genetics, no? Yeah. So, and then I don't want to read out the awards. There are plenty more than what is written here. She is fellow of all the academies. She is a recipient of C.V. Raman Award and Harsharup Medal Award, the unknown ones, and she also has a National Bioscience Award. So I read in the reverse manner because she is a member of all the academies. And the research I can tell without looking at what is written here, because she is the one who works on the meristem generation, the flower meristem, and she has been working on this area for last few decades and to identify the flower meristem generation transcription factors and regulators by doing the single molecule as well as omics based approach. So she's going to wrap up that concept of cellular morphogenesis based on the model of flower meristem. So this is what she's going to do, and she's going to give you a good idea. She has prepared herself to deliver to undergraduate students and postgraduate students. I hope she will come down a little to explain things to students as well. Please, well. Now you can come.
We are not in competition, but I know Usha since she was a PhD student in Caltech, number one. Number two, she's an MS in zoology. Can you believe it?
Biochemistry. BSc in zoology.
BSc in zoology and then MS in biochemistry, and then worked on spicing first, right? And then moved to the plant sciences. Three, no competition, but still.
You won't walk up and down? You will stand here?
No, I got a call on mine.
Yeah, you got it, so that's why I don't feel comfortable. এটা মনে হচ্ছে? এটা মনে হচ্ছে? So, all set to hear our keynote speaker this morning.
Well, first of all, thank you so much to Vice Chancellor, to the former Vice Chancellor, the heads of departments, the organizing committee, Dr. Paul, Dr. Sauri. I'm sure you worked very, very hard in putting together the ideas, the brilliant ideas that your Vice Chancellors have given you. It is indeed a pleasure to visit SNU. It's a new university. But I see the foundations of this university are built on very strong ideals, And I wish you all the best of success in your endeavors. And today, I must specifically thank Dhruboda, Arunda, Maitreyi, and all the Department of Biotechnology and Microbiology faculty members and students for coming here and spending time to listen about the kind of research that we've been doing. Thank you so much, Arunda and Maitreyi, to sort of dwell quite a bit on my background. I'll only say a little bit more to illustrate that just like Professor Thakur was mentioning, he was a drifter. So it's obvious I too am a drifter. And it is by simply this process of being able to move somewhat gear shifts every once in a while and being constantly learning and listening and doing that I've grown. And you can be titled professor. It so happens I'm also the dean, so I have a lot of administrative duties, but I'm always a student. You remain a student for the rest of your life, and that's sort of my motto. Science and nature has so many wonders waiting to be discovered. So in today's talk, I'm trying to pick up on the sentences that Maitreyi said, is on trying to understand how genes control the process of development. And let's go a little bit deeper, and one can study this in the myriads of species that we have on the planet Earth, but let's focus on flowering plants. I'm going to try and use this laser. Everybody can hear me pretty well, right? So that's one of the attributes of being a professor. You tend to speak with or without a mic. Is this too loud? Should I turn off the mic? You're good? Okay. All right. So let's look at development using the model of flowering plants. Now, flowering plants are very highly diverse and species-rich plant group that really are pretty recent on planet Earth. They arose about 180 million years ago, a blink of an eye compared to the archaea that Dr. Thakur talked about. And they last shared their ancestors 1.6 billion years ago. They shared a common unicellular ancestor with the animal kingdom clade. Yet there are amazing questions that has inspired philosophers, poets, developmental biologists, and geneticists when looking at plants as a model. And that is in the vast diversity of the form of their reproductive structures. Those are flowers, right? Any one of you would be inspired just by taking a walk in the garden and seeing what's going on, right? Now, in order to, and this is in fact, been termed by Charles Darwin, one of the most brilliant minds of humanity, who got us the theory of evolution by natural selection and speciation, that the abominable mystery of flowering plants. Where did they come from? How did they disperse and occupy every niche on planet Earth today? And that too within such a short, geological and time span, right? So these were questions that inspired him. They inspired von Gutter, who was a philosopher in late 1700s in Germany. A philosopher, really a polymath, was an administrator, philosopher, poet, writer, everything. In a horse journey going from Germany to Italy, he observed that different forms of plant structures which are coming forward, and he wrote this thesis, The Metamorphosis of Plants, which is reproduced by MIT Press. And many of the things which he conjectured and put hypotheses for required good models for us to be able to understand what are the genetic underpinnings to the models that he actually spoke about and are actually true. Understanding how genes control the process of making a flowering stem and the organs of the flower required the identification of a model system. Sorry, I was putting these slides together and I seem to have missed one important slide, and that is Arabidopsis as a model for all flowering plants. Now, why would Arabidopsis be the model for about 350,000 extant species of flowering plants on Earth? Simply because It has the smallest genome amongst all flowering plants, just 5 times that of the unicellular budding yeast, this nuclear genome. Short life cycle. In 6 weeks, you can go from seed to seed in some of the very fast ecotypes and others in about 8 weeks or so. And the genetics to discover genes controlling any process, whether it is response to heat, whether it is response to light conditions, or development, that is the formation of flowers, can be addressed ab initio by making mutations, identifying mutations that disrupt the process of interest, and then mapping the gene which is responsible for that disruption, and then going to gene function right down to the molecular analysis. So the facility with which one can go do the genetics made this a fantastic model. And I will begin by summarizing about 35 plus years of research, which is focused and drilled down deep to understand how is it that the Arabidopsis flower is made, right? So one just takes a look, observation, to see where do flowers come from. Flowers arise on the flanks of the shoot apical meristem as a simple mound of cells. And this is the young incipient floral primordia, which the German language you'll call it as an Anlagen. So it is equivalent to the formation of the wing Anlagen in Drosophila, or the leg Anlagen, or the antenna Anlagen. So this is the primordia which is going to become a flower. And in the dead center, or the very apex, are present the quintessential multipotent stem cells. So it's very fashionable in the last 10 years or so, since stem cells and multipotency and pluripotencies have been described in mammalian systems. We are so anthropomorphic in our thinking that people forget that plant biologists knew about the totipotency and multipotency of meristematic cells for a century or plus, right? So these multipotent stem cells, which are established in the embryonic shoot apical meristem, divide in a controlled manner, maintain themselves through the lifetime of the plant, and give their descendants, which are pushed to the periphery of the meristem. And in the periphery of the meristem is where organ primordia will emerge. So upon reproductive transition, these primordia are destined to become flowers. So at this moment, they look no different than the multipotent stem cells which are in the inflorescence, but their fate is already determined. Why is this a floral meristem and not an inflorescence meristem that grows continuously and gives multiple flowers? Why does it give only a solitary flower? That's a question to be asked. Secondly, how is its growth controlled? How does it grow in the correct manner to give the right amount of cells, which will eventually give you 4 sepals, 4 petals, 6 stamens, and a single carpel in fixed number, regardless of whether it is the first flower of the plant or whether it is the final flower of the plant, whether it is in plant number 1 or plant number whatever, growing anywhere, Kazakhstan or Bangalore or Kolkata. It will always be the same for that species. So what are the genes that control cell proliferation in the meristem and patterning to give you this invariant pattern of sepals, petals, stamens, and carpels? So young meristem is first indeterminate and multipotent and gradually becomes determinate, terminally differentiated with a specific set of organs which are present in there. Now in Arabidopsis, simply by making mutations which disrupt the process, genes have been discovered. This is now textbook knowledge. If you open any developmental biology textbook, which is at the senior undergraduate to master's level, you will start running into these genes. So one of the hallmark genes in Arabidopsis, which is a critical switch to a flower formation, is the transcription factor LEAFY. LEAFY is a helix-turn-helix DNA binding motif containing transcription factor whose expression starts as a tiny, tiny patch on the flank of the shoot apical meristem, and these cells are now destined to become floral. So the expression of LEAFY portends the future cell fate because LEAFY's expression controls other processes that define the formation of the floral organs. LEAFY itself, therefore, has to be spatially and temporally controlled. LEAFY expression cannot be activated too early So when a young seedling germinates, leafy expression is very, very low or actually repressed, because you want to make flowers after you have made leaves, which are the photosynthetic factory. There's no point in making a flower and attempting to set a seed when you do not have the assimilate to put the energy into the seed. So genes like leafy, which control the formation of the flowers, are temporally and spatially controlled. That network itself is about 80 factors. We are not dwelling into that. Now let's see, what does turning on leafy mean? Once leafy is turned on, it itself is an activator of other regulators, downstream genes, and these regulators are spatially and temporally controlled in where they are expressed on the floral meristem and what do they do. Leafy, once it is activated, activates another transcription factor, apetala-1, whose expression is present throughout the young floral meristem, just like leafy. Leafy and apetala-1 then also are joined by other factors. For example, agamus, which is expressed in the very central part of the floral meristem, and AP3 and pistillata, not shown here, which are expressed like a donut or like a vada. What is this, spatial and temporal control of these transcription factors doing? This allows the assembly of different multi-protein transcription factor complexes. For example, in a certain region of the floral meristem, you will have only the sepalata class of genes with AP1, that is in this outer region. In another part of the flower, you will have the AP3, you will have AP1, you will have the sepalata type of genes, so that region becomes the petal. And in another region, you will have your AP3PI, you will have agamus, you will have the E function genes, and that becomes a stamen. And in another region, you have just the agamus and the carpel, agamus and the E-class genes that gives you the carpel. And within the carpel, you're joined by yet more members, and therefore you get this. So this is like defining zip codes within a particular zonal region. So first you define the zone, you define West Bengal, then you define what is Kolkata, and then within Kolkata, but you're doing this in concentric rings. And each ring is defined by, its identity and differentiation is defined by the formation of these multi-protein complexes, which are all transcription factors, right? All very good and brilliant work done in 35 plus years, really by 4 giants in the field, and Elliot Marowitz, with whom I did my postdoc, and I discovered plant biology rather late in the day. And those of you who are students over here, you can wake up early. Plants are fantastic models to address anything from the molecular to the ecological scale. And Enrico Cohen, who was at the John Innes Institute in Norwich, Heinz Sadler and at the Max Planck Institute in Köln, and we have Martin Corneef at the Wageningen in the Netherlands. So they are the ones who deep dived and developed Arabidopsis as a model to address very many questions in plant biology, starting with flower development. All of that is all very good, but now one goes back to the question of this diversity. While floral organ patterning is highly, highly conserved, that is in all flowering plants, in all angiosperms, you always have sepals, petals, stamens, and carpels. There is a huge diversity in the number of organs per whorl. There's a huge diversity in when flowering will occur. There's a huge diversity on the structure of the inflorescence, that is the flowering stem, and how much it transitions to maintaining its pluripotency versus undergoing differentiation. So a very important a question which is pertinent to mankind, a title over here, is how does crop development occur, and particularly how can we address crop development, flowering in many plants which are of interest for us because our food security depends upon crops. So amongst those 350,000 flowering plants, angiosperms which are there are few numbers which are essential for our livelihood and critical for our caloric intake are cereals, right? And among cereals, no doubt in Asia, the one which tops the list is rice, right? So coming back to Bangalore after my stint as a student doing yeast genetics and then learning plant biology, diving into India, I had to start thinking of what would be the questions that I would ask. What would be the questions that I can hold my own and make a contribution to the field? It was absolutely fascinating to remain an Arabidopsis biologist, and there are many discoveries to be made. But then, one can do a little bit more than that if you're in a new situation. So always see the ecosystem that you're in. What are the questions that you can ask? And that's actually perhaps the hardest thing to do in your career, is defining what would be a question be. Retrospectively, fortunately, I made some right choices. They were difficult choices. It was a difficult decision to say that I will not work with the flagship model for plants. I will gradually transition. And it is people like Ashish sitting right here who emboldened me to say that, we can do things with rice. I had never even seen a rice meristem till I started thinking, okay, this is a model. And why is this a model? It's a model because it is, the smallest genome amongst all cereals. So its genome is only 4 times or 5 times that of Arabidopsis. It's a simple diploid. And at the time that I transitioned back to India, my first paper or so was still Arabidopsis. There were reports coming out that you could actually genetically transform rice much before you could do that for maize. So the second plant which was transformed with agrobacteria-mediated transformation was rice. So I said, okay, if it is possible to do that, then one can ask questions which are rooted in evolutionary questions and development. Genes were discovered in Arabidopsis. The orthologs of those genes seem to be working in floral organ patterning in Petunia, in Snapdragon, 2 very diverse but all eudicot family members. Are those gene members present in the cereals, and are they doing similar jobs? That was the question, which was totally open field. The genome was not sequenced, nothing was available. You have to build up everything from the scratch. So if you're willing to take the deep dive, there are brilliant questions out there waiting in nature to be asked. So I started looking at rice meristems, went to the agricultural fields with friends like Shailaja. I don't know if Ashish, you remember, Shailaja was an assistant professor who wanted to learn molecular biology, so she came to me, and who was I to teach her anything? But I certainly knew molecular biology. She knew how to grow the rice plants. We pulled out the plants from the agricultural field, started dissecting, and found out that actually finding rice meristems is a lot more harder than finding them in Arabidopsis, simply because the shoot meristem in grasses is tightly enveloped by the leaf primordia around it, which makes a pseudostem. So all of that development is occurring in a very, very recessed tissue. So you have to peel apart everything. But yes, once you do all of that, you will find that there are pluripotent stem cells here, there is the peripheral zone, and young leaf primordia are coming. And when the time is right, this shoot meristem transforms from making leaf primordia to one which is now going to bear flowers. But there is an enormous difference in that this inflorescence meristem does not make solitary flowers on its flank like Arabidopsis does. Arabidopsis is a simple raceme, meaning the inflorescence meristem keeps on growing, and in a phylotactic spiral will make solitary floral meristems. Same too for a snapdragon, Antirrhinum majus. But in rice, in maize, in sorghum, in foxtail millet, and pearl millet, what have you, all the grasses, even your simple lawn grass, which is grown outside, the inflorescence meristem goes through many degrees of additional meristem generation through branching. That means the state of multipotency and indeterminacy is prolonged. Prolonged because you generate primary branch meristems, which then make secondary branch meristem, and then finally all grasses will make a short branch, which is called the spikelet. This is the common unit that is there in all grass flowers. The spikelet, what does it contain? The degrees of branching vary, and the number of flowers per spikelet will vary. But the spikelet is the common unit in all grasses, right? Including all the cereal members. You have 2 sterile bracts over here, 2 rudimentary glooms they are called, 2 other bracts which are called as the sterile lemmas, And there are two outer organs, which are called the lemma and the palea, that completely enclose all the floral organs. You will not see the internal floral organs, and it's essentially a closed floret. If you peel it open, then you will see, it's very hard to see over here, so it's drawn out over here, two asymmetrically positioned fleshy organs, which are called lodicules, and these are not present in a concentric circle. The next set of organs are the stamens, which are present in a concentric circle, like in eudicot flowers, and in the center you have a single carpel, which has a bifid stigma and a single ovule within it. So morphologically itself, there are many questions which emerge from just looking at what is happening in rice. Flowering time, of course, is a common question. Inflorescence architecture, how are these transitions of these branch meristems taking place? What is a primary branch meristem? What's a secondary branch meristem? What's a spikelet meristem? All of these structures don't exist in the simple racemes or even the cyme, which is there in tomato. What are the genes that control these transitions of these different meristem? What defines a spikelet meristem? How do you make the lemma and the palea, which apparently are very, very distinct in their structure from that of sepals? They are very thick, sclerenchymatous tissues with a lot of cuticular waxes. They are present somewhat opposite to each other, but not exactly opposite to each other. And the lemma and the palea are actually interlocked. And you can see over here that there is a kind of hooking that occurs. And this interlocking is like a handshake that keeps these floral organs closed. They open only when the lodicules, which are present asymmetrically here, swell. And that swelling is controlled, coordinated with stamen elongation that allows this hook to open briefly for 4 to 5 hours, and that allows the stamen elongation. These temporally and spatially controlled physiological processes allow for stamen emergence and self-pollination. So while the stamen is emerging, it dusts its own stigma and then comes out and allows for some cross-fertilization. But after fertilization, after pollination has occurred, this snaps back shut again because the lodicules collapse. The collapse of the lodicules brings this back together, and that closes this and makes the hull of the seed. So functionally, morphologically, these organs are very, very important. They're very important for allowing pollination to occur at the right time and place, but also for seed setting, because if you fail to open or you fail to close, then you have a defective flower. Now coming back to the genes. So in Arabidopsis, I already gave you this model, which is this ABCDE model discovered by groups all over the world, really. And it is this combinatorial action of these transcription factors that is giving us the sepals, petals, stamens, carpels, and the ovule. Are the orthologs of these genes present in rice? Yes, the equivalent of these genes are present. We searched for them those days, 20 plus years ago, by doing homology-based short probes and fishing them out of the genome, either from genomic libraries or cDNA libraries. Now it's all post-genomic era, as the title of this conference says. You can simply do a bioinformatics search to find the orthologs of genes that you want to. But what was very clear, both from the old way or the new way, that the number of the genes which are the relatives of the Arabidopsis genes are variable. For example, if you look at the A-class genes shown here in the green box in Arabidopsis, contributed by mostly two factors, AP1 and AP2, there are at least 4 factors over here, perhaps more. A lot more genes seem to be contributing to this A function. If you look now at the B function genes, in Arabidopsis there's 2 genes, AP3 and PI. In rice, it's actually 3 genes. There is OSMAD16, OSMAD2, and OSMAD4. So 3 genes are giving you B function versus 2 genes are giving you B function. If you come to the class E genes, which are the coactivators or the mediators that assemble these complexes, these factors are 4 of them in Arabidopsis, and they're completely redundant to each other. These 4 factors are represented by 5 factors in the case of rice. But their domains of expression and their functions have evolved. So I will tell you a tiny bit more, going very deeply into OSMADS1 and a little bit more on OSMADS2. So phylogenetically itself, these 5 class E genes, which are supposed to be related to the 4 genes of Arabidopsis, it's clear that 3 of them have long separated from SEP4. That is OSMADS1, OSMADS5, and OSMADS34. And in this expanded lineage are present only grass members. No eudicot members, no even lily members. So that tells that around the time of the speciation of Poaceae or Gramineae away from the other monocots, this family started splitting and dividing and growing in number. Did they grow in number and share equal functions? If that is the case, then the extra numbers is going to show genetic redundancies, and individual genes, you may not discover a function. Surprisingly, while they have been dividing and generating duplicate members, the genes have acquired new functions also. The loss of function in just one of these genes, that is OSMADS1, done by either a TOS17 insertion or RNA interference. This is a new methodology that came handy for us and many others about 15 years ago, simply by making transgenic plants that express double-stranded RNA to shut down or dampen endogenous gene expression. One can phenocopy what would be the loss of function for the absence of a factor of interest, in this case OSMADS1, or more recently we made a genetic null mutant by homologous recombination, which is actually very difficult to do in plants. Our collaborator Velutambi's lab was brilliant in getting us this allele. Whatever you do, loss of only one member of this clade disrupts your ability to make a determinate flower with a fixed number of organs. This is the normal flower, and this is the OSMADS1 null mutant. What do you get? You get too many floral organs, and each one of these floral organs has also lost identity. So there are 2 things which are gone. The floral meristem should be determinate, meaning the stem cells should divide and give enough cells to make a fixed number of organs. That's gone. So stemness is continued beyond the normal time. Abnormality. Second abnormality is organ identity is gone. Each and every organ is a mosaic. It is somewhere between a gloom, lemma, palea, whatever. Right? Now, where is OSMADS1 expressed? So this required us to do what is called RNA in situ hybridization, to not just grind up the whole tissue and say that yes, OSMADS1 is expressed in flowers, but to look specifically within developing flowers, where is it expressed? So we make antisense RNAs, sections, and then hybridize your RNA to the native RNA which is present. OSMADS1 is expressed all over the floral meristem, and that is consistent with the fact that it is contributing to the identity of the floral meristem. If you don't have this, then the identity of the floral meristem itself is gone. But very interestingly, you look a little bit later on, you see its own OSMADS1's expression itself is spatially controlled. It's persisting only in the little bit of stem cells in the top here, and huge expression in the lemma and the palea. Right? So that means that you lose character over here, you lose the stemless character, you lose lemma palea character, therefore all the organ identities are gone. If those are the phenotypes, what are the genes that it's controlling? Right? To understand what genes it is controlling, we developed antibodies against a segment of OSMADS1, which is specific to that gene, about 20 kilodalton segment of the protein, raised antisera in rabbits, affinity purified those antibodies, and then did chromatin immunoprecipitation in developing panicle tissues. That is this. So remember all that dissection you have to do. You have to collect tissue for 6 months to get enough tissue to do chromatin immunoprecipitation for your transcription factor of interest, which is expressed in that specific domain. Right? So it is technically challenging, but definitely doable. Not something to hold you back. So if you've got the idea that this is what you want to do, you can do it. It just takes more time, but it's feasible. And then after doing the pull down, we sequence the associated DNA, which is present with our protein, and ask where are all the genes that map to the associated binding sites. So the binding sites of OSMADS1, not surprisingly, were around the transcription start site. They were also present far upstream, that is these 2 graphs, but very interestingly, were also within the gene and even downstream. So that tells that OS-MADS1 is perhaps binding to regulatory elements which are enhancers, repressors, likely to be regions which are controlling gene expression from a distance, and perhaps indicating that it is a pioneering transcription factor that binds and changes the chromatin landscape there and changes the gene expression of the associated gene which is present. OS-MADS1 binding sequences are enriched for the CARG element, and the CARG element is the typical element bound by the MADS domain in all MADS domain transcription factors, so that was very nice. But a surprising new discovery was that about 30 to 40% of the binding sites, if you look in the 200 base pairs flanking upstream or downstream, you don't find a canonical CARG element, but you find only this central element, which is an A track, AT track. And this element, without the flanking CG, is present in about 40% of the genomic loci to which OS-MADS1 is bound. And what is the signature of these elements? This AT-rich regions tend to be less stable and less bendable, so that's kind of geometrically very stiff. So in addition to having so this is the average free energy plots for the binding site peak and 200 base pairs upstream and 200 bases downstream. And you can see whether the binding site is intergenic or whether it is in the gene body. It is always the region, the peak region is always less stable and less bendable. Okay, whether it is genic, whether it is upstream, and so on. That tells that the DNA geometry and perhaps the chromatin geometry around that region is also potential signatory for its binding. So now we had this landscape where we have about 3,500 genes of the genome which are bound by OS-MADS1. But the question is, which ones are being regulated and when? Remember now, I have told you that OS-MADS1 is expressed in a large developmental window, right from when flowers are made to when organs are differentiating. In organ differentiation time, it is expressed in the lemma and the palea, but when floral meristms are being established, it's expressed all over, right? So we combined our data sets of ChIP-seq with transcriptome analysis. And transcriptome analysis we did in 3 different developmental stages. One, where early floral meristems are being specified, the very first part of that in situ graph, merging into the second, when organ primordia are coming out, and when organ differentiation has occurred. And then start asking what type of genes does it control in these different processes. So in order to understand that a little better, we first took these 3 developmental pools. That is early pool, which is mostly floral meristems, middle pool of tissues where organ primordia are being established, that is where ABCDE model has been activated and those genes are coming on, and the late pool where organ differentiation is going on, where primordia have already started growing out and final differentiation is going on. So this is like when stamen primordia have to be established, you will establish it, but then you need to make your anther, your filament, your tapetum, and so on, your locules. So that's all going on in the late stage. In the middle stage is when anther primordia are already beginning to grow out, that being an example. Now if you look in wild-type flowers and wild-type floral development and ask how does gene expression change through development in the normal wild-type and cluster the gene expression patterns, you see that there are typical patterns that are expressed. 4,000 odd genes, expression is very high in young flowers and gradually goes down as the flowers are maturing. So this is just the normal pattern in the normal wild type, growing from young floral meristem to organ differentiation to this. There is another cluster of genes whose expression is exactly high in early and middle but drops down in the late. There's one which is exactly opposite to cluster 1, that's cluster 6, very low in early meristems, but as development is going on, it increases. So these are likely genes which are necessary to maintain and establish the floral meristem when it's dividing and growing. These are likely to be the genes which are important for organ differentiation, all of the organ differentiation, right? Now we take this data set and superimpose on that the gene expression data sets which we got from transcriptomics in 3 different tissues, right? So this is very, very data rich, and tells us a lot. So in early flowers, this steel blue bar is about 1200 genes that OS-MADS1 is positively activating. Meaning in the mutant, these 1200 genes fail to be expressed to the levels that they should be expressed. So OS-MADS1 in early flowers needs to activate all these genes to make that floral meristem identity what it is and to make that meristem to grow. What are the typical genes which are present in there? A large number are homeodomain factors. And amongst those homeodomain factors are two key factors, OSH1 and OSH15, which are the equivalent of shoot meristemless and knotted, which are the quintessential meristematic stem cells in all meristems. So whenever you have OSH1 and OSH15 or STM1 and WUSCHEL and all those active, those cells will be stem-like, meaning they will be controlled in their division, they have not yet entered differentiation. So in cluster 1 are present a lot of the homeodomain type of genes. In cluster 2, which is shown over here, again which is high, the patterning genes start showing up and so on. So this is informative on how OS-MADS1 might be controlling early floral meristems, how OS-MADS1 might be controlling late development. So I'll give you a little bit of further analysis over here on early floral meristem. Here we were able to do a little bit of meta-analysis because binding sites of OSH1 and OSH15 were already described by another group. Okay. And we are telling that in floral meristems, OS-MADS1 is controlling OSH1 and OSH15 over here. Not only was it doing so, it was found in the binding sites were found on their upstream. So we show that there is a tight feedback loop between these three factors. In floral meristems, OS-MADS1 activates OSH1, also activates OSH15. These two auto-activate each other, they activate this, making a tight feedback loop, positive feedback loop, which then controls all these other downstream factors. And by doing this, in early florets, you maintain the meristem. And you allow the meristem to grow and organ primordia to be differentiated. So understanding Arabidopsis, phenomenally important. Understanding rice, equally important. And equally important, understanding what your transcription factors are doing in other developmental contexts. Because you can borrow lessons from there and apply it over here. So this is some work which has been done. And one of the students now has developed a very, very tiny little model that in early florets, you have this feed-forward loop between these factors, which over time has to switch and turn into a repression. Because if you keep on having this OSH1 and OSH15 on, the meristem is going to keep on growing and you should get indefinite number of organs, and that never happens. So this switch has to flip from being an activating switch to being a repressing switch, and therefore later in development, OS-MADS1 must be recruiting lots of friends and family and partners in order to switch from an activating mode to a repressing mode. And that's what allows you to have a fixed number of organs in here. I'm going to go to the very last slide, which I will not have time to explain. There's another story that we have. There are at least 3-4 such transcription factor stories. There's another very exciting story which we have on the class B factor, pistillata of Arabidopsis, its relative in rice, which I told you in Arabidopsis, class B genes are represented by 2 genes, AP3 and PI. In rice, there are 3 genes. Pistillata is represented by 2 genes. Again, here we find, while rice might have generated duplicates of the ancestral gene, the duplicates are not equivalent. They are not doing exactly the same job. We found that by genome editing, somebody mentioned genome editing in the introductory remarks, very powerful tool for you to understand gene function. By genome editing, we found that one of the pistillata members, that is MADS2, has acquired a role in flowering time because MADS2 mutants are early flowering, which pistillata is not. So pistillata in Arabidopsis is a dedicated class B gene. But one of the pistillata homologs has gone and taken on a side job. Okay, this side job is controlling flowering time. It's also taking on a more important role in a class B, that it is required for lodicule formation. In MATS2 mutant, lodicule development is abrogated. It cannot be compensated by MATS4 because MATS4 is being normal. Still the lodicule is abnormal. So you have two apparent sisters, cousins as it were, but the cousins are not equivalent, even for class B function. And lastly, we show that these class B function have a role in parthenocarpy, and all of these are very important questions for us from development, but they're very important questions for breeders, because floret opening and closing and fertility controlled with in response to the environment is going to change your seed setting. So early floret opening in climate stressed conditions before you have too much heat will allow gamete viability, will allow your maturation of the seed and all of that. So these are traits which are very useful for breeders. Breeders look at it from just trait point of selection. We got into it based on development, and we think that these genes and the targets that they control are likely of value to breeders also. So this is a developing story. And I've been absolutely, if there's anything that I've been blessed with, any crazy idea that I bring to the lab, however technically challenging it is, I've had fantastic students and postdocs who will take on the challenge, whether it is Ashish taking the train and going all the way to MKU to do particle bombardment in rice to generate transgenics, which he used to do with Kumuda. And this was before even agrobacterium transformation came. So don't let technology be a limitation. Technology is always an enabler. Keep your eyes and ears open. God knows what's going on in any of the schools. You can adopt it. Technology is an enabler. You have to keep your eyes and ears open. But the questions, you have to define it. So I've been blessed by having fantastic students who are all professors and leading lights in their own. I've had great collaborators who worked with me over many years. Uli, very, very helpful in making the transition to doing ChIP-seq with very limiting tissues. Veluthambi in helping us to make the genetic knockout by homologous recombination before CRISPR-Cas9 became technology available to others. Manju, Binay, and Arijit entertaining all our bioinformatics and biophysics and questions, and helping us to build what we have done. And thank you all very much. I forgot the thank you slide.
Hey, thank you, Usha, for such a wonderful talk. It's always been a pleasure hearing you. So if you have any questions, I would prefer to have questions from the students. That is what she's also looking for. Yeah, please. Yeah, someone please.
If we can have the lights there, I can at least see your face.
Yeah, sure, ma'am. A very good morning, ma'am. I have two questions related to the presentation which you made just now. My first question is, can you please explain about the ABC model of flowering in one of your slides, which you told about organ patterning? The first question.
Okay, so yeah. So this, even if you didn't understand, it's not a problem. It's now in textbooks. So if you open Plant Physiology Tays and Ziegler, you will find this explained. You'll also find it in Molecular Biology of Cell by Bruce Albert. So I will give an answer now, but it's quite possible that I may not be able to explain everything. So ABCDE model was based on the observation of mutants. So mutants were found in Arabidopsis and in Antirrhinum, where organ position and organ identity was disrupted. For example, in the pistillata mutant of Arabidopsis, instead of making sepals, petals, stamens, and carpels, what you will get is sepal, sepal, carpel, carpel. That means the mutant fails to make petal and it fails to make stamen. That means the gene which is mutated, the pistillata, is a controller for the differentiation of petals and stamens.
Okay.
Right? So that like this, many, many mutants were made, and through that the model was proposed that there are factors called class A factors whose expression is required for sepal and petal development. Class B factors are required for petal and stamen development. Class C factors are required for stamen and carpel development. And it is a combination of these factors which ultimately gives you the 4 distinct organs of the flower.
Okay, ma'am, for this, which you told just now,
Yeah.
this ABC model is for only for the mutant variants, right?
No, 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 no.
Determination.
The mutant inform you about the wild type.
About the wild type. Okay, okay.
The mutant, because of the mutant, we are able to tell what is going on in the wild type.
Okay.
Right?
Okay, ma'am. My next and the last question is, as you just talked about OSMATS1 transcription factor, so, and you talked about few terminologies, that OSMATS1 determination by chip sequence pull down. So how did you do this? I'm just curious to know about it.
Right. So this is again a technique which can be used for any transcription factor in any species. You can do it in Drosophila, you can do it in, you can do it in worms, you can do it in mouse, and even human tissue.
Okay.
So what you do is you want to understand where is the transcription factor bound.
Okay.
Right. So your genome could be 400 MB, but it may not be bound in every single site. It is going to control only few genes, right? So to do that, you need an antibody against your transcription factor. Your transcription factor is a protein.
Okay.
So you define a domain of the protein which is unique to that protein and not present in any other protein. Raise antibodies against it and use the antibodies to find this protein which is bound to DNA, and then you pull it down. Antibodies can be pulled down because the FC region can be bound to beads.
Okay.
Right, and then when you pull it down from the whole mess of 40,000 genes, only those genes where it is bound, the antibody will be able to pull it down.
Okay.
Then you sequence what is the DNA associated with it.
With the transcription factor binding site.
Exactly, yeah.
Okay, thank you, ma'am. Thank you so much.
All right.
Thank you, ma'am, for such a great talk. There's only a single question that I have. The different organs of the rice floret, how can you justify it evolutionarily?
How can you justify?
In an evolutionary manner, how can we justify it?
Okay, so it has to go back to just like Darwin was inspired about the origins of flowering plants, one has to look at what is the ancestor for all the Poaceae, right? How can we justify? We can justify based on if there is an ancestral Poaceae which we can find either from paleobotany samples or somewhere, and ask how did these organs evolve over there.
Okay, ma'am. Thank you, ma'am.
You got it. So this is because it's the lemma palea lodicula shared amongst all the grasses. The origin of these organs must be when grasses split from lilies. You're getting what I'm telling?
Yes, ma'am.
Okay.
Yes, ma'am. Thank you, ma'am.
OSMATS1, it expression-wise it looks like sepetala but it's a non-redundant because you have so many mutations phenotype you are getting in OSMATS1. So what is the equivalent Arabidopsis? Is it sepetala-like?
It is sepalata-like, but a sepalata 4-like perhaps. Sepalata
But,
1, 2, 3 and 4 are expressed uniformly in all the organs.
So it is also required with the agamas looks like because you have
Yes,
too many
yes,
florets
yes.
over there.
No, that is because it is stemness is continued.
Yeah, I'm interacting with agamas like.
It will control agamas. It
Thanks.
controls agamas.
Excellent.
So it is related distantly to sepalata, but unlike sepalatas, which are uniformly expressed all over the floral meristem, this one is expressed in specific domains.
Okay, as usual, madam, very good talk and intense data set you have shown. With the OSMATS1 chip sequence, it's binding with those poly A or poly T stretch, which is unbendable. Is there a structure already solved of this Matchbox BHLH DNA binding domain with the DNA?
For the MADS domain bound to DNA, it is not known, but it is known for SRF, serum response factor, which is S stands for serum response. MADS, M-A-D-S, M is MCM1,
Okay.
A is agamous, D is deficiency, S is SRF.
Why I'm asking that, it's difficult for me to imagine that the DNA stretch which is not bendable, on that BHLH.
It is less bendable. So Jitendra, you should understand. So the rest of the flanking region is little more flexible. This is little less flexible. It's an AFE plot, so that region is little bit unbendable. And that signature is also there in the carg. Remember, the carg central part is also little bit stiff. So it binds as a heterodimer, at least in that crystal structure of SRF.
Usha, there is a question there.
Madam, it's always wonderful to listen to your talk. I have one question, like some of those MADS box genes like MADS1 or MADS2, in dicot, like in Arabidopsis, they usually are involved in organ differentiation.
Correct.
While in case of monocot, like rice, they are also involved in early part of the floral development.
Right,
Like,
nobody would have predicted. So that's why it is doing their ancestral function.
Yes,
Nobody is saying it is not doing that.
Yeah.
So OS-MADS2 is also controlling lodicule formation, which is the equivalent of the petal.
correct.
But it also has additional functions.
Right. So is it like a kind of a general evolutionary trend that is true for monocot versus dicot, or it's too early to
No, I wouldn't
comment?
say.
Okay.
I'm sure it is there even within the dicots.
Yeah. Okay. Thank you.
You would have gene family evolution even within dicots.
Okay. Thank you.
Most are very quick questions. I think first one is being answered what you said to. Unlike in Arabidopsis, this 5 gene of E-class gene in rice, they are not redundant.
No.
Yeah, fine. And your MADS1 regulates several thousand of genes. You have 6 clusters showing. So is there any evidence of any one of them knocked out and giving a similar type of phenotype? The downstream genes, not the MADS2.
Right. So see, because it's controlling so many downstream targets, a single target may not show the phenotype, may not show exactly the same phenotype as MADS1. It cannot recapitulate everything, right? So downstream to MADS1, there are many others. So double mutants and of MADS1 with other factors have not yet been made. All right. We know that at least downstream to it is one other transcription factor. We published it last year. I didn't show that because we were getting two data. I deleted all those and put the introductory
it's
slides.
fine,
Yeah, so that's BZIP47. In BZIP47, what you get is slightly larger meristem. Organ identity, only stamens are slightly abnormal. So it is accounting for subset of the functions. If you look now at OSH1, which is a downstream target that I showed you, OSH1 is active in all meristems. So OSH1 mutant is embryonic lethal. So we need to have a special allele of OS-MADS1 such a way that shoot meristem and embryogenesis is not affected, but only in floral meristem it is affected. Then we can make the double mutant. Right? Yeah.
Is the chairman allowed to ask one question? Thank you. So first question is, MADS1 is upstream, and then it turns on the toolkit of WOX and all homeodomain proteins to turn on the meristem machinery, right? Then it's common to others. That's why it's embryonic lethal, right? But these managers, like MADS1, who actually control the whole thing, and above that LEAFY.
Right.
The LEAFY and MADS1, LEAFY is the one you said is sensor to the spatiotemporal physical parameters.
That's flowering
the
time.
location. Okay.
It comes under
All
flowering
the
time.
physical, I mean, after all, it's the response to the whole plant and the environment that the LEAFY thing starts.
Right.
Right? And then how does it turn on this MADS1?
You have to call me again.
No, thank you. Thank you. Thank you so much. Okay. Thank you.
so
Let's
the-
put a big hand for Usha once again for this wonderful talk. That's a nice answer. Thank you. We will love to. We will love to.
No, that's simply because LEAFY also is very different.
Yeah, sure. Yeah, thank you again, Usha.
May I now request our Honorable Vice Chancellor, Sir, and Professor Moitri Dasgupta, Ma'am, to kindly felicitate Professor Vijay Raghavan. Thank you, sir. Thank you, ma'am. Now we will proceed for a short break, and we will resume back after 15 minutes. Before that, may I kindly request our distinguished speakers and Honorable Vice Chancellor to be seated here for 5 minutes, and rest of the participants can proceed for the break. Distinguished speakers, may I request you to stay back for 5 minutes, and sir. All other participants can proceed for the break.
disease compared to the set which are naive. So that happened because of the infection memory which developed in the prior inoculated plant, and that leads to the higher level of resistance. So it is identically similar to vaccination and what we call as a systemic acquired resistance. So this SAR or systemic acquired resistance remains active weeks to months and even reported for years. And it also works like a booster. So if you give one booster, two booster, it actually elongates. Like if you have single dose, 1 week, you give 2 boosters, it can last up to a month. So it's all aspects, it works like a vaccine. So in brief, what happens is that you have a primary pathogen. Primary pathogen will generate some signals that can travel to the distant tissue, can go to the apex, and even newer leaves which come up would also get that property that they will be more resistant when you infect second time. So that signal goes and, okay, thanks. And so I'll briefly describe you one specific experiment that we have done long time back. This work we started in 2005, almost when I joined JNU. What we did is we took a mutagenesis program in Arabidopsis and selected a mutant that have a loss in memory formation. So I'll try to explain to you what is loss of memory here. So there are 2 sets of plant. This plant was previously not inoculated or buffer inoculated. This plant was previously inoculated in some other leaf. And here is the number of bacteria in log scale in those 2 plants. So what you will see, the plants which are not infected have almost 10 to 50-fold higher number of bacteria compared to the plants which were infected earlier. You can see the disease symptom. Look at the disease symptom. Here is much less disease compared to this. But if you look into the mutant that you identified by EMS mutagenesis screening, so when you talk about memory mediated protection, we look about the difference between this and this. You see there is no difference between these 2, indicating the mutant fails to generate the memory and to produce memory-based protection. And also look at the difference between these 2 bars when actually they are not infected earlier. And what you will see, there is no difference between them, telling the gene is not involved in immunity as such or not a defense gene. Because if you had a defense gene, if you knock out, there will be more susceptibility. That doesn't happen. Only it generates a memory, and if you knock it down, the memory is lost. So that was a very early work we did in 2000, published in 2013. It's not working. So this is the gene we call as a reduced systemic immunity, or RSI1, that gives SAR. And we did a map-based cloning that actually, thanks to Usha, I learned a lot of that from her. And what we found that it has a mutation from glycine to arginine FLD gene, flowering locus D, that promotes flowering transition.